Standing interest in viral and bacterial genome assembly and analysis, and he was an assistant prof at J. Craig Venter Institute in Maryland before he went to Hunter College. For those of you who don't know about Hunter College, that's part of the City University of New York, which has several campuses around the city, and it's, in my estimation, the lead campus. And Hunter College is a really neat kind of melting pot. It's classifies as a historically black college and university, although I think it's more of a Hispanically serving institution, actually. And 11 undergraduates from Hunter College have gone on to win the Nobel Prize. It's a really neat place. It's a melting pot. It's fun. I love working there. Konstantinos was trained at a place which is a very important place for bioinformatics, and that's at the Virginia Tech in Blacksburg. Virginia Tech has had one of the leading bioinformatics analysis centers in the United States, largely funded by the Department of Defense, and you know, it's a significant place. So he's had very nice experience there, complementary experience at Venter, and then more complementary experience both as a core director and as an associate professor at Hunter College. So I think it's just the kind of person that we are really looking for, and we are anxious to hear about your vision for building the next generation. So Bob, maybe you guys can tell us how many generations there are now. The next generation of bioinformatics infrastructure for next generation sequencing data analysis. So welcome. It's great to have you.
Thank you, Brian. Well, thanks everyone also for coming today. So here's my titles. I think, Brian, you covered it all very well. So
I didn't mention it. I should mention it. Back to the point that it's really tight, 2 blocks away. You know, the founders of 68 and Clarke. 2 blocks away towards the river is the Weill Cornell Medical College. Your laboratories down at the new medical campus. I know you're aware of it. That's Weill Cornell down on York.
It's all the way on the east side. Yeah, that's relatively new. We just moved there in a brand new building, and we have a lot of collaborations with Weill Cornell. So it's been great. Let me see if I can advance this. What happened? Is Alex? Yeah. Spent like 20 minutes testing this. Oh, yeah. I can quit and reopen it. It's a good start. It's revenge of Microsoft. I can get it. Don't run Microsoft on a Mac. I have 2 talks. Yeah, just hit play and keep our fingers crossed. Okay, seems like it worked. Okay, so again, I did my PhD at Virginia Tech. I'm here in the US first time in 2002, and then since then I kept moving north. I spent 5 years at the Clarke Analysis as assistant professor. When I was there, it was right at the time when the human microbiome project was really picking up at the institute, and I've done a little bit of work with outsourcing compute cycles on the Amazon cloud during my PhD, and their computer cluster was running out of compute cycles, so they hired me to do some work in that. Like, great experience. 5 years, unfortunately, was all soft money there. So despite having at my last year there, $2 million NSF grant is called PI. It only covered like 50% of my salary, so I had to look for something else. And then I looked academia. The opportunity at the City University of New York, Hunter College came up. So I moved there in 2014. And through great efforts of our president, we have this collaboration in a brand new building that you see there with the Weill Cornell Medical College. So we've been there for about 1.5 years. And by the way, actually, I always want people to look at this building at the Clarke Analysis you see here. Those are actually colors which match the basis of DNA sequencing. So, you know, Clarke vendor is always a little eccentric, right? So that's a small eccentric. They had big ones. So okay, yeah, I know. If you sit and read there and say of his dog or his name or something, yeah, very probably. Okay, so let me tell you a little bit about my lab. As you can see from the titles, we're a bioinformatics lab. So I hire both staff and students that I train. Some students actually with transition to staff. We do a lot of software development, so we have sort of very heavily software-focused people there. We also, for the bioinformatics pipelines that we build, we have the bioinformatics analysts, and I'll show you a little bit in the next slides how we build those pipelines. You know, we're bioinformaticians, so we usually don't wear lab coats. This was from the official opening, so it looks cool, right? We have an Illumina NGS sequencer, and I'll talk a little bit more about that. And we do all sorts of NGS from genomes and transcriptomes of corn snails to cancer cell lines. The focus of my research, which we'll see more, is development of novel bioinformatics software for large-scale data analysis. And I'm very interested in genomic data mining to efficient visualizations, and even things like getting bioinformatics to work on mobile apps. So, of course, we publish all our software as open source. Some of you might be familiar with the GitHub software repository. So some faculty in this department, they had code repositories there. And when we publish something, we always want our software to be very usable, which is going to be a lot of the theme in that talk. But as part of the usability, I mean, manuals that are published on GitHub, we also put like videos. We have our own channel on YouTube, so we explain like how the software works, you know, features and all those things. The 3 years I've been at Hunter College, I've taught like I brought 3 new courses for programming for computational biology, NGS data analysis, and bioinformatics algorithms with Python. And I was discussing just before this talk with Margit, and I saw that you do a lot of that stuff here, having a strong bioinformatics program. We also do a lot of, since I run the bioinformatics core, we do a lot of specialty workshops, whether it's going to be like bioinformatics data analysis or even wet lab workshops, like how to build your libraries with Illumina kits. And then I have all of my students giving talks. There's a lot of stuff in New York City, right? Like from corporate stuff, pharmas, many different universities, conferences. So I have my students going there all the time giving talks. And specialty programs like the Science Research Mentorship Program. It's a special program to get high school students that can work in the lab. And this is like really top of the students. I actually had 2 students from there. One, she did the research in my lab, and then she got admitted for undergraduate to Carnegie Mellon, and the other one to Stanford. And I also have my students participate even in business plan competitions. One that we do was the entrepreneurship lab, highly selective, 10% entry, because I'd like to think them sort of out of the box and get outside of the scientific thinking, which is all papers and grants and things like that. Okay, so I've been lucky enough the 3 years I was there to have almost $1 million in funding. My startup from the City University of New York, a couple of pilot awards through the Center for Translation and Basic Research that I was hired through, and also since I'm associated with Weill Cornell, one of the pilot awards to the Clinical and Translational Science Center. But most important, 3 months after I arrived, we submitted a grant to NIH, which was, and let me go one step back and say, like, the City University of New York, despite being a good school, it didn't have any, I mean, it had NGS, next generation sequencing, in the sense that people were sending samples out, but there wasn't any sequencer or any bioinformatics cluster in school. So we put this grant in within 3 months that I arrived as a supplementary to the National Institute of Health, bioinformatics supplementary for the CDBR, and we actually won it for $185,000. So with that, what I did was I designed and built a bioinformatics specific cluster. And for those of you who do bioinformatics and are familiar with the clusters here, like the Flux, it's a standard cluster, right? A set of mid-memory nodes with 120 gigs of RAM and 16 CPUs, so we can run like genome alignments, blasts, and things like that. A couple of high memory nodes, half a terabyte and a terabyte of RAM if you want to run assembly or multi-threaded. For those of you again really into bioinformatics, like multi-threaded jobs in alignment, and a data storage array. All those interconnected with a high-speed backplane network. It's actually 2 head nodes for redundancy, and we host that our data center at the Center for Translation and Basic Research. And with the remaining funds, what we did is we built an actual wet lab, sequencing core with an Illumina MiSeq, and then the way we have set it up, it has nothing compared to the sequencing facilities here at scale, but it has all the basic things we need, like a bioanalyzer, QB, PCR machines, and a MiSeq. And then the data we produce from that, they just feed directly in our cluster. So, and with some things I'll show you in the upcoming slides too, we try to make it as easy to use and as transparent. So the PI submit their samples, and then the data appears to an interface on the cluster, and they can immediately go and do their analysis. And you can see here some examples of projects, lots of variability from RNA sequencing, small RNAs, cancer lines, and even corn snail transcriptomes for a project that we had for identifying novel peptides that can be used as pharmaceuticals. So really exciting projects. And a lot of collaborators as well, both in New York City and elsewhere, that we do both sequencing and bioinformatics projects. So overall, it's been great experience. Lots of collaborations, lots of papers coming out. So I've been enjoying it so far. So let me talk to you about a little bit about my research, and then things I've seen like through my experiences and involvement with research building bioinformatics pipelines for NGS in the last few years. So when we think about NGS, we think about like big data and big sequencing, right? Like today I was lucky enough to see the DNA sequencing core. Here's an example of a sequencing core from the New York Genome Center. So we think like hundreds of thousands of dollars investments, a lot of data coming out, dedicated teams to run both, the sequencing, and then with so many data coming out, dedicated teams of informaticians to analyze the data. But there's been a little change in the paradigm the last few years. I mean, the companies do that because they're looking for profit too. But the way I like to say it is like sequencing is becoming more of a commodity. So you have like the sequencer that we have, the MiSeq. So this one costs like $90,000. They have a smaller one, the MiniSeq, which costs $50,000. And then you also have those the Oxford Nanopore, which is like sort of a USB powered sequencer, like really a handheld one. And then the library prep is really getting easier and easier, right? So that's me. When I started there, and we didn't have a couple of other faculty, we didn't have any people to work in the course, so we were doing our library prep ourselves, right? And it actually worked. So if I went in the lab and actually worked, so that tells you it's easy. And the price in, I guess you can multiplex a lot of stuff, especially when you do metagenomics. It's decent, but it keeps going lower and lower. And then, with those instruments, you might not be able to do like whole human genomes, but you have the reference human genome, and then you can do resequencing or transcriptome and stuff. So you can discover a lot of stuff there. And of course, small bacterial or fungal genomes and 16S, a lot of it. That's what those sequences are. So I see the NGS with those developments becoming easier and easier, and then being used more and more as a standard technique in molecular biology. But we have the sequencer to generate the data, and then the way I see it is 3 main problems. One is that, and of course, they all center around the bioinformatics, and one is running bioinformatics software. It needs significant expertise, right? Because you have all these Unix software engineering requirements. The second part is that those sequencers, despite being small, they still can generate a few gigabytes of data, which it's manageable. But then, if say you want to run alignments against the reference genome, human genome index of multiple gigabytes, and then your alignments can run tens of gigabytes, and then those they need to run on computers with 60 gigabytes of memory. So you need a lot of computation capacity. And of course, even after you manage to run the bioinformatics pipelines, then you get like a lot of text-based data. So how do you go about like interpreting those data sets? So I'll talk about that, visualizations and all those things. So a little more detail describing. So when complexity, right? So when you want to run a bioinformatics pipeline, you have 5 to 10 different software components, and all those you might need to compile it from source code, and you might need additional libraries, and all those work on Linux and command line. Then the structure of the pipelines. How do you move data from one tool to the other, and how do you make sure that your pipeline, you follow the right steps in the right order? The interface, you have to run the command line, and also portability. In bioinformatics, we develop pipelines on the command line on a specific system, on a specific Linux system. So once we build a pipeline, how do we transfer it in another system? Because if we were to build something and publish it, how other researchers and other labs could benefit from our work. And just to what I said, to make it more visual, here's an example. So that's the standard RNAseq pipeline with, we call it Tuxedo pipeline, obviously from the names, from the Trapnell group. This used to be a professor at the Clarke Analysis, John Hopkins now. So basically that's the schematic from the paper they published, which has all the tools of the pipeline, and it tells you the steps. Again, that's just a schematic in the paper, but if you actually go around the pipeline, you have to do something like that. Okay. So what about if I told you that there was a way that you could do this pipeline instead of just having a schematic in the paper, have this sort of, let's call it a workflow canvas, and then having the tools as boxes, which you can drag and drop from here, drag and drop a couple of tools, and just connect them, and then create the steps of the pipeline. And this is an actual working tool, which we call a Galaxy. Now, I apologize here, there's a lot of lines between those tools. You have to use your imagination. But we tried before. They told me it's a new project, it didn't work. But what can we do? So essentially, this is the exact same pipeline that you saw in the schematic in the paper, that Tuxedo pipeline for RNAseq. I mean, it's standard pipeline, not anything crazy. My point here is I want to show how we went from that sort of schematic in the paper and actually sort of living, breathing pipeline, which I don't know, what do you think? Like, could somebody who doesn't have any experience in bioinformatics, if we gave them this interface, they could actually get the paper and then drag and drop the tools here on this workflow canvas and connect them with lines together and assemble a pipeline, which might be working, or you know, after a little tweaking, might be working, right? Instead of running everything on the command line like I showed before. And that's what we do at the Bioinformatics core at the City University of New York, because what I found was like, and I'll explain the technical stuff here in a little bit, but before that I want to say that what I found in the 3 years I was there was that we were a small university, a small core. There was a lot of interest in NGS, but we wanted to find ways how we can sort of teach a man to fish, right? So teach our faculty and our postdocs how to build bioinformatics and have the right tool in the interface, so then they could pick up themselves a lot of the work that's needed in bioinformatics, instead of everything having to go through us, and also like as an educational tool. And the choice for that was the Galaxy web platform. So essentially, what we were doing in the developers in the core was they were writing those XML files, so the extensible markup language. So essentially, what that is that you describe the command line tools, the inputs, the outputs, the parameters, the data types, and then what the Galaxy does is like reads this file, and then it renders the tool in the workflow canvas. And then it's going to render the tool with all the inputs and outputs, and also when you try to build a workflow and connect the tools, it's going to check what the data types that you specified that the tool they create as output, and what this tool gets as input. So if they're compatible, then it's going to let you connect it, or not if they're not compatible. So it really takes away a lot of the sort of the manual work in bioinformatics. And this, again, I apologize, the lines don't show, but this is like, again, an RNAseq pipeline. I'm using an example, but it's a more complex one, because the first one that I showed you, it was kind of a simple pipeline. You know, it just had like 2 conditions, and then 2 data sets, 2 reads as input per condition, like forward and reverse. This has like 8 data sets as input, having 2 technical replicates. So that's more of a real RNA-seq pipeline that you would run if you would need the replicates. And then if you would see the lines in here, so essentially the first stage of the pipeline is some fast grooming, how we call it, for doing some, let's say, quality check of the reads. Second one is the alignment to the reference genome, the top hat. And the third one is the cufflinks, which after you assemble your reads, essentially gets the, after you align the reads, gets the aligned reads and assembles the transcripts. And then further on is the, what is it, the cuff diff, which does the differential expression, and the cuff merge and the cuff diff, the cufflinks and the cuff merge. So if you would see here, you would see like there's so many lines between those tools, and you would think like each of those lines, it would be like one operation on the command line, right? So you would want to run, you know, you read data set through the groomer, second read data set to another groomer, third read data set to another groomer, so you can check the quality of each data set. And each of those runs would be one command line, so one line between here. And then each groomed data set, you would want to run it through the aligner, through the top hat, and so on and so forth with this tool. So imagine like how many command lines you have to do. Each operation, you would generate one output, which you would then have to get and feed as input to the next tool. And things would get like a lot complicated. Of course, you can write shell scripts and things like that to do that. But then again, shell scripts are usually more understandable to the person that wrote them, and not anybody else. And then there's so much, if you want to teach bioinformatics and you want to teach about pipelines and those things, there's so much you can do on the command line. Where this approach here, I think it works much better. And just to give you some more examples of pipelines, we have this one. It's not, I mean, it's for bioinformatics data sets, but not bioinformatics analysis. So what it does is basically data transformations. again, like I mentioned, when you have bioinformatics pipelines that just generate text data sets, and what we end up unfortunately doing a lot of times is like getting them in Excel and doing a lot of data sorts, column cuts, and those kind of things, which is one person does it and then they don't remember what they done. Where here we captured the process in a pipeline. So essentially that gets the raw pipeline output and generates custom reports based on what the faculty were collaborating requested for. It might be reports sorted by p-value in the differential expression, locations in the chromosome, gene names, and all those things. Also, this was actually from a huge RNAseq project for rat. They have what they call the mine discovery rats, a project funded by DOE. So they wanted to see, like, when they expose them in TNT smell, which genes change the expression. And then we also inserted links in those reports, which actually link to the genome browser on the RATGB. And then this was a great project because then they presented the results to the DOE and they got the funding. So we continue the collaboration with them. And of course, the simplest case with the Galaxy platform, you just if you want to run just a simple tool, again, that's all the command line parameters of the tool as they are entered in Galaxy by iterating the XML that we had defined in the backend. And that's what the software developers in my group do a lot. And just to explain a little more, what we have here in one column is the list of all the tools, which you can click in one and you get the tool interface, and then you can enter the parameters to run it. Or if you are in the workflow canvas that you saw on the previous slide, you can drag and drop those here and create your workflows. That's the Galaxy history here. So every data operation you do, so essentially if you upload the data set or you had a data set uploaded already, and you run an operation, by operation I mean running a bioinformatics tool or workflow which generates output, all those appear here. And then the way we have set it up, when we run a sequencing experiment, that gets, as I mentioned before, streamed on the cluster. And then under the researcher's account, the sequencing data appears here. So they can go by themselves and run tools and run pipelines on them. So what we think we did is that we made it really simple for them to be able to do sort of basic and even advanced bioinformatics through this interface. And then that's our cluster again. So the Galaxy sits on both our head nodes. I want to show one here. So essentially, when researchers get the sequencing, the data is here, and then they appear in Galaxy. They can go around workflows on the cluster, and then whatever workflows they're done, the cluster queuing system automatically finds where we have like free resources and submits the workflow to a node with a free resource. So that was what made a huge difference for me running a small bioinformatics core and also running my research as independent research as a faculty to save a lot of time while doing the bioinformatics with limited resources. So hopefully so far I convinced you that with the Galaxy, we can take away all this sort of complexity of the bioinformatics pipeline, like the Linux sort of stuff. With the workflow canvas, have the structure of the pipeline in a more organized way, which we can understand. We can use it sort of like a presentation tool or information exchange tool when we do collaborations in bioinformatics with PIs. The interface is friendly, so someone without any command line expertise can run pipelines in Galaxy. Again, we come back to the issue of portability, right? So I build my pipelines in Galaxy, works on the head node of my cluster just fine. But what about someone who gets a sequencing machine and they don't have the access to my cluster, right? In another institution, and say they're a small institution which they cannot, they don't have the funds to buy all this infrastructure to have or hire somebody to build that stuff from scratch. So that's the portability issue. So let me introduce a new concept for you, which is the concept of a virtual machine. So what is a virtual machine? One way to say it in one sentence is that a virtual machine is a fully featured Linux server in a single binary file. So a virtual machine would be that here. So and a way to understand this is that if you think about the cloud, right? So essentially, what is the cloud? And I'm sure you all have heard like the cloud is something like the cloud at Amazon, for example, where you can go like pay per hour and servers, right? So what Amazon has is on their data center, say they have like a huge computing machine here with 250 gigabytes of RAM, and you go say like I want to rent for 3 hours a machine which has like 125 gigabytes of RAM. So what they do is that they fire up virtual machines. And that virtualization layer, I guess they have their proprietary one. There's many open source ones too, which you'll see in the next slides. It sort of allocates the resources, like half of them, 125 out of 250 that you asked, in your virtual machine. So you get your virtual hardware operating system, usually Linux, and then it might have applications or not, depending like what virtual machine you started, or if they have like some pre-configured ones. And when you use those virtual machines, whether you do like a remote desktop or Galaxy, or if you do command line stuff and you log into those virtual machines, it has no difference from a regular server. Like you would sort of, for those of you who've done like secure shell or let's say login in a remote server, and remote being like from here to say the Flux server that's on the data centers of the engineering or wherever this cluster is. That's the same thing. If you log in a virtual machine on Amazon, it wouldn't have any difference. So this publication that you see here, it was the first bioinformatics virtual machine that I built. We released in 2010. The paper came out in 2012. That had bioinformatics tools, and it was up on Amazon Cloud by Linux one. Great work, a great paper, got a lot of exposure. So this one, it came. Our Cloud by Linux virtual machine on the application layer had more than 100 pre-configured bioinformatics tools and pipelines. And then, yeah, so the whole idea was that how can we, for places which they don't have any previous bioinformatics expertise or access to bioinformatics experts, we can make bioinformatics easily accessible. So, and that was when I was at the Clarke Analysis, so we had this consortium with a small university in South Africa, the University of Limpopo, and Clarke Analysis Genomics Network. And imagine this university was so small that this computer room was basically the only one they had for like library research, teaching like anything from business classes to like the students going there doing their homework. And what we did is that we got those virtual machines, and I was talking about the cloud before, but if you notice, there was a virtualization layer. So there's actually desktop virtualization applications as well. One being, for example, the VirtualBox. VirtualBox, like you can download it, like you download Skype and install it. Then once you have that, you can get our virtual machine, which is what we did here, get it on your computer and have all those bioinformatics tools pre-installed. So we went there and then installed the virtual machines in all the computers. So essentially, we converted that lab to have very easily, without going through any manual effort to install tools, to have bioinformatics functionality. We taught some workshops, and then, of course, since it was open source tools, we left it there for them to teach their classes. And this is some of the examples. Again, this paper, the Cloud by Linux, was back in 2012. So some of those might be more older tools, but those of you who do NGS analysis will recognize them. But again, it's been great, lots of collaborations, pipelines built in the virtual machine, and through collaborations and papers we got out of them. So what I want to show you here is sort of like the latest incarnation of this project. So what we did is that besides like the standard Cloud by Linux tools, we also installed the Galaxy, which we use a lot, and then a visualization suite, which I'll show you in the next slide. And then we moved from VirtualBox to Docker virtualization. Some of you might know it. It's much lighter, much better performing, and we have a paper which we just submitted. So the whole idea is that wrapping all those tools in here, and what this paper, if somebody's interested, I can send you the draft. What this paper describes is that how we can wrap complex pipelines into the virtual machines, and the pipelines, they're all built in the Galaxy workflow canvas, and I actually had students who were coming in without bioinformatics expertise, and they were firing up the virtual machine, getting the Galaxy interface, and then I had them like curate the literature and find pipelines, and then my software engineers were like building the components of pipelines through the XML to expose them in Galaxy, and then those students without bioinformatics expertise, they were following the schematics in the paper and assembling those pipelines in Galaxy, and then we were saving that virtual machine. And that coupled with the virtualization interface, what it does is that when you run the pipeline, the output of the pipeline is fed into the visualization, which we'll see a little more in a little bit. And you know, that stuff with the virtual machines, okay, I can run it on my desktop, I can access it, but it can get a little technical, right? So what we have in this paper is sort of a meta script. So like you would run, say, a Perl script, which does some basic bioinformatic analysis on the command line. What it does is that it abstracts all the processes of running the virtual machine. So you can go run a script, and what it's going to do is going to fire up the virtual machine. It's going to ask you where your data is. So you might have like, you know, gigabytes of RNAseq data. It's going to get those inside the virtual machine, drive it through the workflow engine of Galaxy, run it through the pipeline, and the output give you the output of the bioinformatics pipeline loaded in the visualization, ready for you to see. And we'll see some examples of those. So hopefully, you know, I convince you so far that we can sort of, with this approach, attack the first problem. Somebody who doesn't have any bioinformatics expertise in their group, they can get the virtual machine and run our pipelines. Of course, the second problem remains that bioinformatics tools require computational capacity. So this one turns to be the easiest one. This is what I mentioned before, the Amazon cloud. So again, you can go on their data centers and run servers, which are virtual machines, which have no difference from servers. By the hour, they have multiple data centers, West and East Coast, Brazil, Europe, Asia Pacific, and Japan. There might be, I think they have one in South Africa too. They keep popping up, like some of them. And the cost of the virtual machines, it varies based on the capacity. If you request a large one with 250, I think that's probably like higher now, while the price might stay the same. Request 250 gigs, you're going to pay like almost $5 per hour. Where if you just get like the smallest one they have with like 4 gigs of RAM, you pay like 5 cents. And then that's the storage cost. So all our virtual machines and those things, they're up on Amazon. So if you just have a laptop, you can go there, fire the virtual machines, upload the data, and then do the analysis there and just pay for what you use. Another option is, because sometimes with Amazon, you have to use a credit card and stuff. I mean, if you know the right people inside and you do like a lot of business with them, they can invoice you. But nowadays, especially for the example here of the Mac Pro, I looked at the numbers, and the teraflops that you get with the Mac Pro, essentially the same with the fastest supercomputer that you had on Earth in 2003. So in that box, and you can get it for $42.99, and it has 64 gigabytes of RAM. So you can do a lot of bioinformatics on your desktop, or if you are more like a Linux person, you can install you can buy one of those Dell servers which we have in the lab, and those ones we run like RNAseq pipelines. One whole pipeline to complete it takes about 20 to 24 hours or something like that. So you buy one of those computers, you download VirtualBox or Docker, which again it's very easy to install. Like you say, you would install Skype, and and then you pull the virtual machines, and you're ready to go. So with that, we think that we can help like small labs that are getting into sequencing. They don't have access to big bioinformatics cores, to analyze their data. And again, the whole idea was that, like I mentioned before, right? I have students or even postdocs who are, they're not really in bioinformatics, but they get in some sequencing, then they can go curate the literature. We have the workflow canvas. They can assemble the pipelines there. And then once you have the pipeline assembled and the virtual machine safe, then you can distribute anywhere. Like for example, here you have all these amazing computational resources, and I haven't met with any of them, but I'm sure like they would be flexible to install some virtualization layer if you want to run there those virtual machines. Of course, you can always go on Amazon, and then if you have your own server in the lab, you can run the virtual machines there. The key is that we remove that all initial effort to install the pipelines, or with the pipelines that we have pre-installed in the virtual machine, or if one pipeline is not there that you need, you can assemble it very easily on the Galaxy interface. Okay, so I told you the problem B would be easy. Like find computation capacity is easy. Now, let's say a more difficult one. So like I mentioned, you run your pipeline, you get a bunch of data. So how do you interpret those, right? And when we think about, let's talk a little bit about visualization. So when you think about visualization in bioinformatics, you think about something like that, right? I guess we've all seen like that, the traditional Gmod genome browsers, right? So what's the problem with this one? And I'm sort of telling you this story for one visualization application that we developed and published, and I think it's pretty innovative the way it does things. But the way that was driven was that I looked at those visualizations. I was like, we have this complex software stack, right, where it's like the Linux servers, and then you have databases, Apache web server, and then on top of that, your web app, PHP, for example, or Perl, CGI. And sorry, I'm getting too technical here, where your application runs. So essentially what happens is that, say you open the genome browser, and then you want to move a little, 5,000 bases on the right or whatever to see your gene. You click the move to the right button, and then there's a whole like command triggered through that whole stack, and then excuse me. So it goes in the database, fetches the data, and then sort of redraws that whole thing, which is very inefficient. And then most of those visualizations are static images. There's nothing like touch enabled there, especially now we live in a mobile age. So and of course, when you have a stack like that, think about how complicated it might be to maintain it. And then you have things like the Google Genomics and the Google platform, where everything is like very modular. There you can like everything was controllable through an application programming interface. You can tell the API for short. So you can tell the API to go like fetch data from or put data in a cloud database. You can tell it to run a computer in the App Engine, and so on and so forth. So so what the Google Genomics does, their goal is to store genomes on the cloud. So you can actually store your genome for $25 per year. And it's a big effort through the Global Genomics Alliance. You can read all about it in the links. But seeing that, we thought like, why couldn't like applications in bioinformatics be this way, right? And I don't like, I'm a very open source guy, so I don't like, the big corporate players and stuff like that. But they do amazing things, right? Or at least they give the good example. So back to the visualizations. So if you read the New York Times, you've probably seen a lot of that stuff. It's actually JavaScript in a JavaScript library called D3. So we were like, okay, let's use this language which creates those cool visualizations, and let's get like very nice graphics like those, and just drive our visualizations from data that are on the cloud, like Google Genomics, for example. And yeah, that's the components, I guess, I mentioned. So we use D3, which is a JavaScript library. We use HTML5. So, and that's a publication, and I'll show you a little more about the visualization. So our visualization runs purely in the web browser. So essentially, it's a file that you load in the web browser, and then it comes alive. No data stored in the backend database. It pulls data live from the Google Genomics Cloud, Dropbox. Say, for example, you have a collaborator does some sequencing for you, puts the runs a pipeline, puts the data on Dropbox. You can just pull it directly from the Dropbox on your web browser that's running our Visualomics Explorer, and then visualize them. So the reason we went that way, like I mentioned before, of course, first to go away of that complex software stack that's under the surface, and the second was like, the second I guess was the cloud, and the third was like, we thought that the browser is becoming the computer, and that's true. Like if you think the Google Chrome laptop, right? Which basically when you get one of those laptops, what you get is like a full screen browser, and which you can never minimize, I guess. And technologies that drive that, one is of course is JavaScript, and then also the HTML5, which is the latest definition of the language, which actually has multi-threaded computing built into it. So you can do like parallel computing in the browser, in a sense, not in a sense, in reality actually. Our goal was to see how we can do something like that in bioinformatics with pure in-browser computing. And this is some of the example data sets of Visualomics Explorer can handle. And I will show you also, given that it was in JavaScript, there's a framework called PhoneGap, so you can get JavaScript applications and convert them very easily to mobile applications. So very easily we could get a touch-enabled bioinformatics visualization. And then, since I mentioned, what you're going to see, I have some videos coming up, how it connects to the Google Genomics Cloud. This is some of the data sets Google Genomics has available. So you have accessible through our browser. You have access to all those data sets. Okay, so let me try and play those videos. So that's the mobile app. So basically, it just scrolls through all the visualizations, the RNAseq. We'll see some of those in a little more detail. This is one that does phylogenetics. So you can upload phylo-XML. This is actually the Google Cloud. It was zooming in some variants. In the interest of time, I'm going to spend a lot. This is on the right. It was the variants in the different chromosomes. You can arrange the number of pins per chromosome that you want to have your variants. And you can see all very easily touch enabled. Yeah, when you click one of the bins, it actually gives you the list of all the variants. I'm talking about the right one that are in the bin, and then you can scroll through them, and each variant links on the UCSC genome browser. And then on the left is the phylogenetic tree for metagenomic data. That's the phylogenetic tree visualization if you run a lot of 16S. Again, because this all runs cloud-based, that's why it's enabled to run on the phone, right? Because you just pull the data live from the Google Cloud. But don't do that on your cellular. Do it on Wi-Fi, okay? So there might be a lot of data. Yeah, yes, yes. Yeah, you can upload like just regular data sets, Dropbox, Google Drive, and then Google Genomics, which is their Genomics Cloud, and it just pulls those live. Okay. Okay, so this one I want to skip the first part. I just shows like the data sets. So this one is variants, DCF files or BD files, like for example, say if you did a ChIP-seq, apologize, the contracts is pretty bad. So when you first load your data sets, it just groups it default set of bins. You can select the chromosome. Time, like scroll it forward a little bit, and you can arrange a number of bins. When you click, let's see, so he's going to select. Those videos are the ones which we have on the YouTube as part that they link in the publication as part of the tutorials. So he selected different chromosome, and then on that chromosome, can adjust the number of bins. Yeah, so let's see live here. So instead of 10, put like more bins. So since we increase the number of bins, it becomes more dense. And then you can click on a bin, and then it can give you the list of all the variants in this bin, and also link out to the genome browser. Yeah. So here's the list of variants, and it's going to scroll down, and each of those is a link. So it shows you the variant, and then it's a link to the genome browser. And then the same you can do with BD files. Okay, so I'll show you a couple more. So this one is the RNAseq. Again, it shows, I'm going to skip that, it just shows the dataset. So the dataset that the RNAseq does is the ones, because we were doing the CapDiff, the Tuxedo pipeline a lot, so it visualizes CapDiff output files. So that circle here, it shows you, so it has a default p-value, and it shows you how many genes that met that p-value for differential expression, how they distributed per chromosomes. The two lines here are the two conditions, and you'll see, like, as he goes to the, so we don't have sound, as he goes to the chromosome, the whole idea to that was to pick up really quickly, like genes which have like differential expression between the conditions. Obviously, those are not very far apart, except this guy maybe. This one is FPKMs. This one is not, while it's linear, it's not actual positions on the chromosomes. This one is actually lines in the file. So let's see. Yeah, so that's what it does. And again, like I mentioned before, so again, the contrast is pretty bad. That's zoomed in this area. If you were able to see here, you'd see like a lot of peaks, right, which is like all the lines in the data set, and you zoomed in this area, and that's what you see in large here. But again, the goal was to have interactive visualization of those type of static bioinformatics data files. Let's see what I have here. Yeah, you can change the p-value. You can click on the circle on a specific chromosome. It's going to get into that. If you click on that, it gives you like the specific information, the chromosome, the gene, the FPKM, and the details. So essentially, the whole idea is like you can really scan through it and pick up, from the lines visually very fast the differential expression between the two conditions, and then click with one of them and get more specific information. And then the last one, last but not least, the Google Genomics Cloud. And again, that whether you run on the phone or in the browser, it connects live to the Google Genomics application programming interface. So check the reference assembly, the data set, and then within that 1,000 genomes, specific genome, coverage, chromosome name, position, and then it's going to pull the data sets. From the data set, it's going to pull the information for the variants. So essentially, it shows you the aligned reads, and then stop it. Okay. So so this was the videos. Okay, so just 4 more slides to conclude this talk. So moving forward, I mean, I still believe that you know, we need to do strong collaborations with biomedical scientists for development of novel bioinformatics solutions. I mean, close to my research and the stuff I like to do is strategic development of bioinformatics infrastructure for large-scale NGS data analysis. But even closer to you know, my interest is that how can democratize bioinformatics so we can make them easily accessible for basic research, medical practices, and clinical and field applications. And what I show you here is not the collection of palms, but sort of where I think the bioinformatics field is going. And this is actual product you can buy, a USB-powered handheld sequencer from Oxford Nanopore. I mean, those guys that had the USB sequencer, they thought like, why can't we do a sequencer which is like powered by Lightning port? Essentially, that's a sequencing chip, is a nanopore based chip. So essentially, the DNA goes to the nanopore and it measures voltage changes to identify the bases. And they thought, why don't we scale this down? This is a handheld computer, which I'll tell you in the next slide how we use Illumina MiniSeq. And I mean, the nanopore, they do really amazing stuff. They have those microfluidics devices, which is in beta, which is for very easily preparing your library. So essentially, you would throw a DNA sample in here, and that's programmable also through an application programming interface in a software library. And you would write some code and do your library preparation, and then load that in the sequencer, and you're ready to go. And that's like that's sort of an alpha testing, like a sort of one-shot library prep they have. And does anybody recognize which is this one? It's the actually the spot where they put the finger in Gattaca. So the reason I put this here is that because if you see all that stuff computing in the palm of our hands, also sequencing in the palm of our hands, that doesn't look very far, right? So yeah, so we're not exactly there yet, but I think all those technologies are going to make like a huge change, like the way we do like biomedical checkups. I mean, if you go the Illumina way, there's a lot of wet lab stuff involved in the sequencer, but then Illumina, they have the advantage of data analysis, one being the base space cloud, which they try to put on site now. So you buy those computers and then they just push up applications for data analysis from their cloud. And then the Oxford Nanopore, they have like good stuff going on with those handheld library prep and sequencers, but their software is still at a very early stage. And then what we do in my group is that we're looking how to leverage software and hardware technology like the handheld computers I showed you and the handheld sequencers to make bioinformatics easily accessible as any other laboratory tool. We're actually developing a compute cloud which similar to the base cloud, but they can deliver open source bioinformatics apps through the virtual machines directly in your sequencer, and that's an actual screenshot because the MiSeq, that small sequencer, has quite a powerful computer in there, so you can run some stuff. So I envision it when this when this is published, that somebody would get our apps and be able to like you know, just operate the instrument in their labs and do the analysis right in the instrument. You know, like single-use sequencers. This is what you saw before. The handheld, it's called the Intel Compute Stick, and the processor that this has is actually the same like the 12-inch MacBook. So it's pretty powerful computer. You can run single chromosomes, RNAseq alignment there in a few hours. But those are going to keep getting stronger. So you know, I see those things like going together and giving us the next wave of bioinformatics. And we're also exploring how we can use that sort of offline with a big battery here. So our compute stick and nanopore sequencing, completely off the grid sequencing and running data live at the same time. And I think you know, while we still need funding from the agencies to develop software and pipelines, I think they're going to start switching, and you're going to see like funding calls for more applied technical research, and that's where we would like to come with that stuff. Of course, there's the option, because it's getting more and more difficult to get R1 grants, where we can start, and we'll explore that, start a small company and put an SBIR grant, which is a good mechanism to fund like a postdoc or something like that, an engineer. I saw here you have at the University of Michigan amazing stuff like the M Kickstarter, which for type of research like that, it can help like building a company towards SBIR and bigger grants like the M track. So if you had an SBIR and you ran out of funding, you could use that sort of an intermediate. And of course, corporate funders once you have enough traction. So I think, just to conclude, there will be a time when you will go do a blood checkup and you're going to get like deep sequencing for, say, for example, circulating tumor RNAs, or capture like tumor cells with structural variations or aberrant gene expression in your blood cells. And sequencing will only become simpler. The cost will decrease and the throughput will go up. But the data analysis will be the bottleneck, and I think and that's what I want to do, keep doing as researcher, making bioinformatics simply accessible as any other laboratory tool to user-friendly software and scalable data analysis. And with that, I'd like to thank you for your attention, and if we have like a couple of minutes for questions. Mm-hmm.
All this wonderful capability you have, what kind of research would you like to do with it for yourself?
So definitely, yep. So definitely, I think that developing more efficient pipelines and more efficient in the sense not only on the computational aspect, like to run efficiently and run fast, because the data sets will keep getting bigger and bigger. More efficient in a sense to include as much of the data that is out there, because it's kind of a cycle, right? So the more sequencing we do and the more projects we have, the more we can get those and feed them back to aid the discovery and get more precise, like whether we're talking for discovering variants or discovering gene expression or anything for like spatial temporal stuff. Like I know here you're very interested in the high C and the chromatin and those things. Like the pipelines have developed so far as like RNAseq, exome sequencing, variant discovery, ChIP-seq. But I'd be very interested to get into those type of pipelines. And I think there's going to be a time where we're going to have like multi-omics data, right? I think that's kind of the next step, because the sequencer, again, I think 5 to 10 years, you're just going to get like one use sequencers like the nanopore or something else, right? But I don't know, there might be instruments which are like multi-omics, which can measure proteomics and variants at the same time. So that's where I think it's a big challenge for the pipelines that we can develop. So yeah. And then I think also like as a medical school, and as I was reading through the pages, there's a lot of push for commercialization and entrepreneurship. I think those type of devices that maybe they could find a way, of course there's all the regulatory hurdles there, in a doctor's office, right? Or even, I know the funding agencies like NIH is interested in that, and sort of like getting the sequencer, like the sequencing off the grid, and the bioinformatics off the grid for developing countries, or like disease outbreaks or disease monitoring and things like that. So I think there's a lot of potential there as well. Pathogen detection, yeah, live, real time.
So my question is, in your many collaborations and with papers written, how far do you go analyzing the data, and when do you hand it off to your collaborators?
So you mean at the bioinformatics and genomics core?
Yeah.
That we have? Yeah. So essentially, For example, say we do an RNAseq project. Our pipelines, the way they are set up now, and even with the virtual machines, that latest paper that comes out, you can essentially feed them the raw read files, and then they can generate preloaded visualizations with the data sets. And it depends on the collaboration. Like I had collaborations where even the faculty, say, does cancer research, RNA-seq of cancer cell lines, they sort of embed the postdoc in my lab, and then the postdoc is sort of on the data analysis, and then they can work very closely with the software people in my group, and be in that cycle where they get analysis, they get the interpretation and stuff. But I mean, my group is a bioinformatics group, so we wouldn't be doing work where we would be writing, say, a paper in cancer and making statements about what, say, that variant does in cancer, right? This is something that comes through collaborations that I do. But I would do like, the heavy lifting, then 99% of the 99.9% of the bioinformatics analysis.
I really like your presentation and the idea of doing sort of sequencing in the palm of your hand and democratization of bioinformatics and make it easier and simpler. So the question that I have is, have you thought about data, storing the data and sharing the data? And the sort of second part of that question is, we all know that metadata are almost as important as the data themselves. So have you thought about conceptually at least how to handle these issues?
Yes. So for the metadata, I completely agree. And the way I approach this is like I try to capture the knowledge. Say, for example, for variants inside the pipelines. So if we were to build, say, a handheld sequencer with a handheld computer that you can go off the grid in a developing country and say, like, you sequence people to tell them if they have variants which will give them cancer, right? I will capture the knowledge from the database for those variants already in the pipelines, so it would give you the answer as when it detects them, if they are in there. Of course, again, it's all like the regulatory stuff and how you can make claims and everything, how you could bring this in practice, right? So. Yeah. Yeah. So essentially, that's the genomics core which I run. So what we do is that I mean, don't think we do like any cell culture or things like that. In the new building that we are with Weill Cornell, it's all like an open collaborative space, like all glass labs. So, for example, when I work with a faculty that wants to sequence cancer cell lines, right? So in their lab, they grow those lines, they extract the RNA or whatever that's needed there, and then they bring us this extract RNA, and then we just feed that in the we're a small course, so we use like the standard Illumina stuff. So we just feed those in the Illumina kits, and then we do the library prep, and then we put that in the sequencer, and then we pick it up from there and do all the bioinformatics. So that's the 30% of the wet lab part. And I also have many times that, because, even for people who do wet lab, like put on their CV that they actually prepare libraries for sequencing, that's a big plus for like later employment opportunities and things like that. So we have people from other labs coming in and contribute that wet lab time as well. So this morning we talked about the importance of in the course of the report, we talked about that a little bit. Did you share that with a lot of people? I don't remember what we talked about. Yeah. Okay. Yeah. So that's kind of also what I showed with the Galaxy, right? Sort of like the best practices. And you know, that's what I found too, that had a great value for me, like being like constraining the resources that I have, and finding a way where you know, I can teach people how to use those tools. And of course, you know, when you have an easy to use interface and a very accessible interface, that can make it much easier. And then sort of remove a lot of the workload we have, instead of, you know, having everyone and saying like, okay, run this, run that. When we have something like that, then we can remove a lot of the workload. That's the one you were referring to, right? So, okay. Thank you.